Factors affecting enzyme activity. Temperature and pH. As you can see in the diagram, for every enzyme there is an optimum temperature and pH on which it is highly active. If that optimum pH or temperature is altered, enzyme will lose its activity. In case of lower temperature than that optimum temperature, enzyme may be preserved, but if temperature is high, the enzyme will lose its activity because of the denaturation of the protein. Concentration of substrate. You can see in the diagram, concentration of substrate initially rises the enzyme activity until and unless all the active sites are binded, but after that the enzyme activity is declined because the number of substrate is more than the number of active sites of the enzyme. Also, there are certain chemicals which when bind to the active site of enzymes, they inhibit the activity of enzyme. These are also known as inhibitors. In certain cases, these chemicals or inhibitors, they are very similar to the substrate. Hence, they are known as competitive inhibitors. And when these competitive inhibitors, they bind onto the active site of the enzyme, now substrate cannot bind, hence enzyme loses its activity. Classification and nomenclature of enzymes. Oxidoreductase or dehydrogenase, which catalyzes oxidoreductase reactions. Transferases, it helps in transfers of the terminal group. You can see here. Lyases, they helps in transfer of groups with the formation of double bond. You can see this reaction. Isomerases, it helps in conversion of any isomer form. Ligases, it helps in attachment. Cofactors, proteinaceous part of enzyme is known as apoenzyme. Apart from proteinaceous part, enzymes may have three kind of cofactors which are required for its activity. These are prosthetic cofactor, coenzyme cofactor and metal ion cofactor. Prosthetic cofactors are tightly bound and are responsible for the enzymatic activity. Coenzyme is bound only during the time of catalysis and later on it is dissociated. Metal ion example is zinc for carboxypeptidase is also bound to apoenzyme to increase or to maintain the catalytic activity of enzyme.